Hello, good day. Thank you for giving me the opportunity to share my work on modeling tissue-dependent fibrosis. This work was done at EMBO Barcelona by the Haas Lab. Let me just tell you a little bit about what we did. So briefly, fibrosis has been implicated in about a third of all global fatalities. And many of the hallmarks of fibrosis have to deal with myofibroblast activation, deleterious extracellular matrix remodeling, as well as compromised tissue structure and function. When you look at the intersection of fibrosis with cardiopulmonary mortality, we see that cardiac disease itself is the leading cause of deaths worldwide. And TGF-beta, or the transforming growth factor beta, has been implicated as the major player of fibrosis in many tissues. However, the process of fibrosis differs in the heart and the lung, which there are changes in inflammation, in the extracellular matrix stiffness, as well as in collagen synthesis and degradation. However, we don't fully understand why this is, and some people have attributed to reduced understanding of these tissue-dependent disease models to our current preclinical models, which mostly use 2D cell culture and animal models that tend to not account for tissue scale complexities as well as interspecies differences and doesn't resolve in tissue-specific fibrosis. Seeing that both the heart and the lung are highly vascularized tissues, our lab decided to explore what the role of the vasculature would be in a fibrotic-like setting and see if we could replicate tissue-specific fibrosis in vitro. Our lab has previously published papers where we developed perfusible microvascular tissues using UVECs and stromal cells, so those are human umbilical vascular endothelial cells, as well as lung fibroblasts, and to show that we can develop tissues in a chip, in a 3-chamber chip, encased in a fibrin gel that can be perfused with different media. For this particular project, we're interested to see if we could replicate cardiac-like and a lung-like environment. So we adapted our previous protocols using human-induced pluripotent stem cell-derived endothelial cells and co-cultured them with or without cardiac or lung fibroblasts. And what we then saw was that we could form these perfusible vessels and that these fibroblasts also interact greatly with the vessels themselves. Interestingly, we wanted to understand how this affected vessel morphology and function, extravascular matrix remodeling, as well as to see if we could reproduce tissue-specific fibrotic response. Early on, what we saw was that the morphology was influenced just by the co-culture of these cells, of the fibroblasts with the endothelial cells, and we saw changes in the area coverage, the diameter, junction, as well as branches, with a reduced diameter significantly seen in the cardiovascular co-culture, the cardiac fibroblast co-culture. We then wanted to see how it would impact the endothelial barrier integrity. So we looked at how certain fluorescent molecules would go from the inside of the lumen to the extravascular space. And we saw that the cardiac co-culture also increased the permeability greatly, and our measurements were comparable to other published results. But we didn't understand why this was so. So we then looked at the inflammatory profile and saw that the cardiac co-culture was more inflammatory-like, which could have resulted in the increased leakiness or the increased permeability noticed in our systems. Additionally, we wanted to explore how this affected the extravascular remodeling. So we did FRAP experiments in the extravascular space, as shown in the circle outside of the vessel, and we looked at how long it took for the bleached region to return to its original intensity, and that gave us an indication of stiffness. We also saw a correlation with a difference in the cardiac versus lung co-culture. We then tried to explore this by using nanoindentation, and what we saw was that tissue mechanics were altered by stromal cell types, and that there was an increased stiffness in the lung co-culture itself, and that these measurements were correlating with the measured diffusivities itself. Seeing that differences were observed in the microvascular tissues we created, we then wanted to explore TGF-beta-induced fibrosis. And what we did was we did a series of nanoindentation experiments at low and high concentrations of TGF-beta, and we saw what differences in fibrotic stiffness related to what had been reported in the literature. We also saw that smooth muscle actin expression as a marker of myofibroblasts were in line with what measured stiffnesses we had, as well as the matrix metalloprotease activity also correlated with the measured stiffness. In summary, we saw that tissue mechanics were altered by TGF-beta. There was an increase in stiffness with the lung co-culture, a decrease in stiffness with the cardiac co-culture, and as well as a smooth muscle actin and MMP activity correlating with the measured stiffness. I want to say thank you for the team that made this possible, as well as the funding agencies that made this possible.